So my research is really very peripheral to the area of antimicrobial resistance. So what you'll hear from me is an outsider's perspective of how we viewed the problem over the last couple of months after the invitation to this meeting came. Now, we know that, and we've heard from earlier speakers, that there are established mechanisms of resistance. These include efflux pumps, destruction of antibacterials, and the mutation in the antibacterial target. What becomes important to remember is that these are natural evolutionary responses that bacteria have to their environment, and this is going to be a continuous process that they keep doing over the course of their lives. What becomes interesting is the concept of persisters, and this is something I think which is not so well addressed in terms of thinking about antimicrobial resistance. Persisters, as we know, are those that are killed slower than normal wild-type bacteria in the presence of an antibiotic. But this persistence is not a consequence of any mutation that they have, because you can find ready interconversion between persisters and normal bacteria over multiple passages. Therefore, this is an aspect of bacteria that we really are trying to understand now at the molecular level, and could arise purely by epistatic mechanisms or through stochastic differences in the population of bacteria that we're dealing with. And I think this is something that we're going to have to think about when we talk about antimicrobial resistance. Interestingly, this figure that I've taken is from a collaborator of mine in my institute who's actually interested in cancer and resistance of cancer to various types of drugs. And they make direct correlations between antimicrobial resistance and cancer chemotherapy, showing that the underlying mechanisms remain the same, whatever the complexity of the organism or the disease that you're dealing with. Just providing a little bit of information, which again I was able to glean while I was reading about this whole topic. This is the picture of antimicrobial resistance in poultry farms. This is in North India. A variety of poultry farms were sampled here, the soil within the chicken cages, as well as in the area around. And this includes agricultural land. And what you can see here is that a number of bacteria are resistant to a number of different types of antibiotics. This is really what's meant by this getting into the environment. And as a consequence of this, what we're dealing with is this rather horrific statistic which I saw and which has been alluded to in the morning, and that is the incidence of sepsis in newborn neonates which are admitted to a Delhi hospital when they were actually born outside of the hospital environment. More than 20 to 30% of these neonates die from sepsis, and when the organism that was causing their death was identified, they were found to be again multi-drug resistant. Now, where were these children born? In villages in the north, around the poultry farms which were using the antibiotics. Now, there's no direct correlation because we don't have such detail, but the effect of this is pretty severe. So what are the mechanisms by which antimicrobial resistance is achieved? We've heard a lot about genetic reasons or mutations in targets for antibiotics causing resistance. But what struck me as very interesting are two papers that I came across here. One, this paper that appeared last year in Nature Communications, where the investigators exposed salmonella to very low doses of antibiotics, sub-MIC levels, and monitored the development of resistance of these bacteria to such low levels of antibiotic. This is somewhat similar to what you'd have in the environment, where you had some low level of antibiotics being present in the soil, say around the poultry farm. What they identified were that mutations occurred in 5 genes, which were absolutely unrelated to the development of resistance to streptomycin. But the bacteria were very, very resistant. They made mutations in each of these 5 genes and found that no single mutation contributed significantly to resistance, but when all 5 were present in the bacterial population, significant resistance developed. So we're now dealing with the organism being able to develop mutations in genes which we do not know or did not classically believe as being contributory to developing resistance classically. The second paper that I'd like to highlight is from a young investigator of mine. He happened to be a former PhD student in my laboratory, and he intermittently exposed E. coli to rifampicin. So he provided it for a little while, took it away, provided it again, took it away. This is again perhaps mimicking an environmental condition that might be seen by bacteria. And what he identified was that there were mutations in a gene which was important for flagella biosynthesis. These resistant bacteria to these intermittent exposures of rifampicin adopted a small colony variant phenotype. This, of course, would have contributed to less permeability of the drug into the organism. But again, we had an example of a mutation in a gene that was completely unrelated to the regular gene that's associated with rifampicin resistance. And I think this is the complexity of antimicrobial resistance that we're going to have to deal with. And it may be insufficient for us to analyze bacteria purely by existing mechanisms of mutation for us to be able to appreciate the complexity of antimicrobial resistance that we're dealing with. Of course, I can't go away without talking about tuberculosis, where we may have had a little bit of research interest. This is a very nice review written by Gagnon sometime, and as mentioned earlier by Timothy, any mutation in a gene that is a direct target for the antibiotic usually results in fitness costs. However, there could be epistatic mutations which now allow the bacterium to overcome this fitness cost and therefore grow at a level which is normal or similar to what the wild-type bacterium does. And again, just coming from our own research interests in looking at the regulation of cyclic AMP in mycobacteria, we just overexpress this metallophosphodiesterase, which is important for degrading cyclic AMP in mycobacteria. And when we overexpress this and we looked at gene expression changes in wild-type MTB as a consequence of this overexpression, one of the genes that went up is INIA. Now, this gene was downregulated in our strain, but in a recent article that appeared in Nature Genetics, where they analyzed thousands of multi-drug and extra drug-resistant tuberculosis, this particular gene was correlated with antimicrobial activity to a high level. The gene I work with has nothing to do with antimicrobial resistance to date. But if overexpression of this particular gene results in alterations in gene expression of those that are involved in antimicrobial resistance, we have a very complex network and systems way of thinking about things. So what do we need right now? It appears to me that we need new technologies to be able to address this particular problem. Here's one example, again from a colleague of mine in the Institute. He has taken the approach of trying to use phages to be able to deliver antibacterials, especially in pulmonary infections. This particular paper, which appeared in Nature Biomedical Engineering, talks about being able to coat particles with phages and deliver them into the lungs of patients suffering from influenza and Pseudomonas infection. These phages were specific for Pseudomonas, and therefore they were able to prevent the killing in rodent models of mice being susceptible to Pseudomonas. Is this one way by which we can now try and look for other alternatives, where the ability of the bacterium to mount a response against the phage may take slightly longer to do than it does in terms of being able to direct it against an antibacterial? What about reducing antibiotic usage? Are there ways by which we can deliver antibiotics to cells such that the levels that are required to bring about effective killing are much lower than what is usually required? This is the work of Professor Shalini Gupta from IIT Delhi. And what she has tried to do is to combine liposomes containing an antibacterial along with a drug which targets specifically cancer cells. Her aim was to be able to kill cancer cells, but also treat infection that sometimes occurs in these tumors. And by doing this, she was able to lower the level of antibiotic required, as well as lower the level of the anti-cancer drug in order to be able to kill these cancer cells. This is what I mean by new innovations coming from a space outside the classical microbiologist or biochemist approach. So what is the way forward? We've appreciated the fact that a lot of what we're facing today is human intervention. And so in the case of the Anthropocene, is this a new time of challenges that we're going to face, new challenges, both from bacteria, animals, and humans? And I'd like to argue that apart from One Health, we're dealing with one world now, because the problems of antimicrobial resistance can no longer be restricted to countries such as India or China. But because of travel of people across the world, this is going to affect every single nation in the world. From experience, evolutionarily, we know who will win this battle. It's a pessimistic note, but we're certain and almost sure that bacteria are going to be able to outwit us at some point or other. Therefore, we need to have an understanding of new mechanisms by which antimicrobial resistance emerges in genes that appear to be completely unrelated to drug targets. And this means that we're going to have to address the question far more insightfully than we are right now, where we're just trying to detect antimicrobial resistance by mutations in well-known and well-studied genes so far. What do we need to be able to do this? We need to be able to do rapid genome sequencing. And this is allowing us to be able to identify mutations in genes other than the classical ones that are involved in antimicrobial resistance. We need rapid phenotypic screening, microfluidic approaches to be able to look at the rate of bacterial growth rather than having to look at it in our regular culture approaches that we're using in laboratories. And of course, needless to say, hopefully some of the targets that are identified could result in the development of new antibiotics. And finally, mathematical modeling of drug resistance. This is an excellent paper from Terry Ha's group in UCSD, where they looked at the fitness costs associated with bacteria developing resistance and how there appeared to be a bistability in the ability of these bacteria to grow, such that one could predict the way these bacteria respond to antibacterials in future. I'll close here. Sorry, I skipped the next talk. I'll close here, but as an outsider, I think what we need are a lot more innovative approaches and new disciplines coming into thinking about antimicrobial resistance in order to be able to take the thing forward.